Today, we begin talking about one of my favorite topics in biology. Evolution is the glue that sticks all the puzzle pieces of nature together. In this unit, we will examine the evidence that supports the idea that all species share a common ancestor. We will talk about mechanisms for evolution like natural selection, sexual selection, and genetic drift. We'll discuss different species concepts, and we will learn how to create diagrams called phylogenies that show evolutionary relationships. Actually, we're going to start with these diagrams because they are really important. Imagine that you love learning about animals and have decided to collect a few hundred dead ones and pickle them. That might seem odd today, but naturalists for over 2,000 years have been doing just that. Curious to understand them, they decided to place organisms into logical groups. This is the field of systematics, and people in this field are called systematists. A systematist might be interested in morphology. What is this organ for, and how does it work? They might also collect DNA sequences to compare different organisms. Hippo DNA is most similar to whale DNA. They would describe new species, taxonomy, classify them, and give them a name, the field of which is called nomenclature. I skipped over something important. How are species classified? Obviously, you don't want one category with frogs and mice and another with pigs and chickens. Mice and pigs are mammals. They share a more recent common ancestor. They should go into one group together. But how do we know? By building phylogenies, the field of which is called phylogenetics. The first person to come up with a classification system for life was, of course, Aristotle. He divided all life into humans with their rational soul, animals with their sensitive soul, and plants with their vegetative soul. In other words, you are either thoughtful, able to move, or just able to grow and reproduce. Since humans can do all three and animals can do two, there is a clear hierarchy. Within animals, there were vertebrates with red blood and invertebrates without red blood. Within vertebrates were categories for mammals, four-legged egg layers, amphibians and reptiles, fishes, birds, and whales. Yes, Aristotle was unsure what to do with whales. Invertebrates could be hard-bodied or soft-bodied, worm-like or plant-like. It wasn't perfect, but it was a good first try. It wasn't for another 2,100 years that Aristotle's theory was seriously revised. Swedish botanist Carl Linnaeus came up with our current classification system in the 1700s. It was similar to that of Aristotle, with categories within categories. He called these categories taxa, and they included kingdoms, phyla, classes, orders, families, genera, and species. So when I say hummingbird, I am referring to the family Trochidae, which includes 361 species. And hummingbirds are birds in the class Aves, which are vertebrates, animals, and eukaryotes. Linnaeus' system was a major improvement, not only because his classification was more refined, but because he decided that each species or kind of organism deserved a single Latin name. In fact, it was a binomial, a genus and a species, always italicized or underlined by convention with capitalized genus. Each species is defined by a specimen that was examined by the namer. Humans were called Homo sapiens, and Linnaeus described us by examining himself. So in that sense, we are all derived from Linnaeus. Rats, wolves, dogs, and all living things were given a name. Some, as you can see, were named with a sense of humor. Now we had a language for discussing life, which brings us to phylogenies, also called phylogenetic trees or cladograms. We must build these trees to identify relationships between species. In this phylogeny, you can see that cats and leopards are more closely related to one another because they share a branch. Wolves split off earlier. The names of the higher taxa and some traits have also been added to this tree. But how do you build this tree? You first need data, which can be comparisons of anatomical structures or DNA, and these can come from living, extant, or fossil, extinct organisms. Fossils are particularly useful because they represent nodes of the tree, where the branches come together. These are the ancestors. Fossils are like photocopies of the hard parts of things that lived a long time ago. If an animal dies and is buried quickly, such that oxygen is absent, with enough time and pressure, the hard parts will be replaced by minerals. Once that happens, a permanent fossil is created. Fossils are only formed in sedimentary rocks like shale and limestone because other ways rocks form would destroy the fossils. The detail can be truly remarkable. Imagine how we would view the world if fossils were not formed. So, we have some data. How do we create a tree? Organisms most closely related to one another should be most similar. We share 98.8% of our genes with chimps, 94% with baboons, and 84% with dogs. Apes are also anatomically more similar to one another, and all primates have opposable thumbs. So you take these measured traits and put them into a table, labeling presence and absence as 0 or 1. A computer program reads the table to produce a tree based on shared similarity. It is similar to how a line of best fit is created for graphs from a bunch of points. Of course, things get tricky. If you wanted to create a tree from cats, dogs, and humans, having eyes would not be informative. We call that trait a plesiomorphy because it existed in the ancestor of all species of interest. Similarly, being bipedal is not informative because only humans do that. This is called an apomorphy. These traits don't tell us about relationships. What we want to find are synapomorphies, or shared derived characters. An example would be long canine teeth, which indicates that cats and dogs are closer to each other than to humans. These traits may also be referred to as being homologous. The external anatomy of arms, flippers, and wings may look different because these animals have adapted to different environments, but the internal anatomy is remarkably similar because, of course, they evolved from a common ancestor. Evolution does not always make this process easy. When two unrelated organisms are under similar selective pressures, they evolve to look very similar. For example, Australian sugar gliders look very similar to American flying squirrels, which also look a lot like flying lemurs from Southeast Asia. The problem is these animals are not closely related. This is convergent evolution and has caused a lot of misery for early systematists. But DNA does not converge the way anatomy does, and if you use enough characters, you can figure out what is going on. Convergence happens. We see it happen often, actually. But finding the right tree shape, or topology, is possible despite this. A phylogeny is really a form of hypothesis that is revised over time based on added evidence. For example, figure A is an older hypothesis for how mammals evolved, and figure B is a more recent one, based on a much larger data set. So Xanartha, in green, which includes sloths and armadillos, was once thought to be the first group of placental mammals to split off, but now we think a group that includes elephants and sea cows split off first. These relationships may seem trivial, but it helps us better understand how organisms respond to environmental change. The simplest explanation is always the most reasonable. We call this concept parsimony. So birds must be more closely related to mammals based on this one trait. But we would rather use hundreds of traits for comparison. DNA is great for this. For example, let's take a look at this matrix of genetic similarity of humans, fungi, and animals. Based on this data, what is the most parsimonious phylogeny? When we group organisms into categories, our focus is on clades. A clade includes all members of a branch, including the common ancestor. Clades are sometimes called monophyletic. We like monophyletic groups because they are the most logical. Again, you don't want a group to contain an unrelated mix of species. A paraphyletic group includes the ancestor and not all descendants. In this example, deer and hippos would form a paraphyletic group because whales are not included. If whales are included, we have a monophyletic group, and we call this group the order Artiodactyla. A polyphyletic group is also bad because it excludes related taxa. In this example, whales and seals form a polyphyletic group because deer, hippos, bears, and dogs are not included. For example, vultures are a polyphyletic group that evolved by convergence. Vultures in the Americas are most closely related to storks, and African vultures are most closely related to eagles. And reptiles are paraphyletic because they do not include birds. Birds are clearly nested within reptiles. Now you try. The group in yellow is the kingdom Protista. Are they a monophyletic group? Because Protista is paraphyletic, a new system was created to get rid of the use of this term. Instead of 5 kingdoms, we now use a 3-domain system that includes bacteria, a clade called Archaea that includes bacteria-like organisms thought to be the earliest forms of life, and Eukarya, which includes all protists, fungi, plants, and animals. With this system, all groups are monophyletic. Before we can use phylogenies for problem solving, we need to go over a few more things. Branch lengths represent time, so longer branches indicate a species has existed for longer. In this phylogeny, which species went extinct the fastest? The branches end at taxa. They could represent species, but they could also represent larger groups like birds or lemurs. The branches meet at nodes, which represent ancestors. The closest relative is a sister taxon. So the sister taxon of A is B. C is the outgroup because it is at the base of the tree, the first to split off. When building a tree, you want to include an outgroup to determine what the ancestral state of characters was. In this example, the lancelet gets a score of 0 for all traits, and anything new that evolves gets a score of 1. Thus, we can build a tree that links all organisms together into one giant phylogeny. Of course, it is easier to zoom in on one branch like these horses in the family Equidae. From these trees, we see that horses evolved primarily in North America, spreading to Europe across the Bering Land Bridge. They started off small, the size of a small house cat. Their leg bones fused to run faster, their teeth changed as they dominated grasslands instead of forests, and some, like zebra, developed stripes, which reduced the number of attacks they get from biting flies. Every character has a story of adaptation and refinement. Remember multi-gene families from the last unit? We can also examine how genes evolve using phylogenies. Replicating a chromosome, for example, will create 2 copies of each gene that can evolve in different directions. This has happened many times and is thought to be of great importance. A speciation event causes copies of genes to be in different species. These are called orthologs, and they have the same function. A duplication event can create a second copy of a gene with a different function. These genes are called homologs. Homologs allow genes to evolve without harm to the organisms. One of the great discoveries made through phylogenetics was the mitochondria and chloroplasts have DNA similar to living bacterial species. This led to the endosymbiosis theory that plasmids were once free-living bacteria that were swallowed by other bacteria and became symbiotic mutualists. This must have happened a very long time ago because all eukaryotes have mitochondria. We can think of this endosymbiosis as a form of horizontal gene transfer, which, by the way, is a bit of a problem for evolutionary biologists. Like convergence, it obscures relationships. Some worry that it is so common that to understand the history of life, we need to untangle a serious mess. Luckily, data suggests it isn't that bad. Because mutation rates are consistent within a clade, we can use the number of changes in DNA as a measure of time. This is called molecular clock theory. And because we have different types of DNA that evolve at different rates, we have multiple lines of evidence to support the hypothetical timing of events. For example, based on this phylogeny, how many millions of years would it take for a 20% difference in protein coding DNA, exons within nuclear DNA? I want to end with my head on this side of the screen. Also, I want to end with one practical example of how useful phylogenetics can be. David Acer was a Florida dentist accused of infecting 8 of his patients with HIV. Based on this phylogeny, how many patients had HIV that was derived from the dentist?
Why does it matter to know how organisms are related to each other? There are 3 broad reasons: diversity, origins, and directions. Let's check this out through an example. By placing these species into a phylogenetic tree, we can see that ants branched off first, followed by the wasps, and then finally bees. The tree tells us when each group originated in evolutionary time. Finally, we can place traits onto the branches of the tree to tell in what direction the organisms are evolving. For example, bees have a pollen basket, but is this because all groups leading up to bees did, or is this a trait that is unique to bees? Placing the pollen basket on the tree, we can see that the only groups with this trait are the bees. And so we can be confident that the evolution of a pollen basket is unique to bees and not inherited from a common ancestor that they share with ants and wasps. Phylogenies give a context to an organism and its characters that allow us to effectively study its life history, ecology, and behavior. Recent advancements in genetics and computer science have made the field of systematics more exciting than ever, allowing scientists to look at and analyze data in ways researchers 100 years ago or even 60 years ago when DNA was discovered couldn't even dream of. But nothing beats looking at specimens under a microscope. Large insect collections like the Cornell University Insect Collection we're in now are better than Disneyland because they contain insects collected from around the world. This collection has about 7 million specimens and is actively adding more all the time. The specimens are pinned, labeled, organized, and arranged in cabinet upon cabinet of drawers. Visiting researchers and in-house scientists utilize these specimens to investigate the insect tree of life. Regardless of what a scientist studies, collections are there to make it possible. Insect collections and collections that hold other organisms are essential for documenting the nature and pattern of biological change. Since new specimens are added every day, a collection acts as a time capsule that stores a slice of a region's natural history for future generations. Today, we take advantage of past collectors' efforts to investigate how populations and individuals change over time in response to human activities. Collections are indispensable in determining which organisms are native and which are invasive to a region, and also how populations change due to climate over time. A great example of this is the Rocky Mountain locust, which we have here in the Cornell University Insect Collection. These locusts went extinct in the 1880s as a result of both climatic changes and human activities. The best part about collecting insects is that you don't need a PhD to do it. The next time you're taking a walk or are just outside, peer into some bushes and see if you can find some insects. While looking at them, ask yourselves, why do they look the way they do? And what was the evolutionary trek that their ancestors had to take to reach to their current forms today?